Hello and welcome to this Nature Research custom media webcast titled Multimodal Tissue Imaging and Machine Learning to Advance Precision Medicine. Spatial Profiling of Human Tissue for Cancer Research and Diagnosis. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Raresight. Effective treatment of cancer and many diseases is increasingly dependent on a precision approach, in which the quantification of molecular features for individual patients guides their treatment plans. However, cancer diagnosis and staging are performed primarily through the examination of biopsy and resection specimens. These methods provide insufficient molecular insight to guide the use of targeted and immunotherapies, even when we understand tumor genotypes. Fortunately, the last few years has seen the introduction of new methods for performing high-plex imaging of histological specimens at subcellular resolution. When processed to extract single-cell data, these promise to combine the strengths of histopathology with deep insight into cancer biology. This webcast will describe progress in the development of new instruments, reagents, and machine learning algorithms to further these benefits. We'll begin by hearing from Dr. Peter K. Sorger, the Otto Kreyer Professor of Systems Pharmacology at Harvard Medical School. Peter leads a multi-institutional effort to advance the basic and translational science used to develop new medicines, create novel drug combinations, and identify responsive patients. He'll be speaking with reference to the characterization of immune landscapes in primary melanoma and predicting tumor progression in colorectal cancer. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to Peter.
Thank you very much, and thank you to Nature Portfolio for putting together this webcast. And as you just heard in that introduction, I'm going to tell you today about recent progress in using multicolor, multimodal tissue imaging to advance both research agenda and precision medicine. So let me just start by giving you a disclosure of my financial relationships. Critical to this particular presentation is that I'm on the board of directors of Glencoe Software, which in the background created some of the open microscopy environment software we use, Raresight, the sponsor of this meeting, and NanoString, which makes spatial transcriptomics instrumentation. So as you just heard, our concepts about precision medicine really postulate that there are going to be two components of those. One of them are going to be a measurement of a disease state. We think of that particularly to be true in cancer, where many of those disease state measurements are genetic, but it's broadly true of any kind of diagnostic modality. And that would then be combined with an optimal therapy in order to provide every patient with the right drug at the right dose at the right time. And what's noteworthy is that the relative investments in these two arms are completely out of balance. And so there's a very active, and I think we all understand, critically important industry building new medicines, but the measurement of disease state has remained relatively smaller, about tenfold, according to these slides from IQVIA, with relatively small numbers of new tests per year. And among those tests, the great majority of them, particularly in oncology, are genetic tests. However, if we actually look at the type of testing that's determinative of disease and actually ends up advancing both the determination of tissue type or tumor type in this case, and the treatment plan, we'll see that actually histopathology is really the dominant diagnostic modality. So these numbers, which are still informal numbers coming from comprehensive cancer care centers in the United States, suggest that genomic testing actually impacts 6 to 8% of all diagnoses. When it does, it's of course critical. Think, for example, the EGFR mutations in lung cancer or the BRAF mutations in melanoma. But histopathology, in contrast, is impacting essentially every single diagnosis and treatment plan. And what's remarkable If you go to a busy histopathology facility in a large hospital, which could process hundreds of thousands, potentially even millions of specimens per year, is that the methodology remains fairly classical, nearly 100 years old, and comprises individual histopathology technicians and pathologists looking at glass slides and then taking that information in their minds effectively and making the critical diagnosis. And that is making a slow transition to digital pathology, but still focused primarily on the classic imaging modality of transmitted light microscopy and hematoxylin and eosin staining. And you see that just in that little picture. We'll see many more of those images in a moment. And that gives you this sort of classic pink-purple view of cell and tissue morphology. So there's been quite a lot of interest, and the topic of today's talk is using new spatial biology approaches, both for cancer research, but also for diagnosis. And we commonly distinguish between two types of biomarkers that might be used. So a clinical test would, in this view, involve an image and a biomarker derived from it. One of them are prognostic biomarkers, and that's would forecast the natural progression of disease, and the other one are predictive. And the most advanced of those are actually companion diagnostics that are used in combination with a specific therapy. There is broadly more interest in predictive biomarkers than prognostic, but it's worth remembering that treatment plans are still very substantially impacted by the former. And the critical distinction from a utility perspective is a well-defined connection between a measurement of some type and some essential clinical decision. And that decision could be to treat with drug A versus drug B, perhaps not to treat at all, to go advanced into surgery, radiotherapy, et cetera. So those are the sort of space in which we would see spatial biology play out. And in the latter part of this talk, you'll see actually attempts to develop, at least in a model setting, a prognostic biomarker that would impact a treatment plan in colorectal cancer. So I'm going to start by trying to derive 6 lessons that have come from about spatial profiling and high-plex tissue imaging in particular. And then we're going to apply those to an attempt to do spatial profiling at scale for these diagnostic applications. And these 6 lessons, as you'll see, sort of impact the way we think about both the analysis and the instrumentation necessary to move from a research setting into a more demanding setting of human patients and human diagnosis. And almost all of this work that I'm going to describe was supported by the National Cancer Institute Human Tumor Atlas Network program. And you can see a link there from Cell, an overview of the program that was put out at the very beginning. And the HTAM program imagined the creation of an atlas of online data in which you would have the analog of what we see in the Cancer Genome Atlas, TCGA. So in TCGA, focuses almost entirely on gene expression, copy number, mutation. There's a little bit of protein data as well, but it's very much focused on cell intrinsic processes. In contrast, HTAN wanted to look at this at the level of cells and tissue, enumerating cell types, programs, interactions, and then place those into the larger geographic context of tissue. And I think you would all appreciate that this is sort of also what's happening in single cell biology, where we're moving from dissociative methods to integrated methods in which one is able to get spatial information as well. So the lessons here are drawn from a couple of papers that have been published. I'll just show those to you. You can find them all. They're all gold open access papers, so they'll be free and immediately available. And the first published earlier this year was a 3D atlas on interactions in colorectal cancer. The other one was a spatial profiling in melanoma at single cell resolution. And then finally, of immediate impact to today's discussion is a paper that you can get access to in bioRxiv and is now accepted and in press and should be out in a couple of weeks, I believe. Also gold open access and has the following authors. And this refers to some instrumentation we developed as part of a small business grant from the National Cancer Institute in conjunction with RareSite, which, as I mentioned at the beginning, is the sponsor of this webinar. The initial 6 lessons are actually not going to come from that development pathway, but in fact from a method that we developed some time ago, and the reference here from eLife, and we call it cyclic immunofluorescence. And so this is sort of emblematic, I would say, of the most common way of collecting high-plex tissue imaging data today. And the idea is that lower plex data is collected between 3 and 5 antibodies at any given time, or 3 and 5 fluorophores. That is then imaged, and that process is repeated, followed by a registration and stitching process to generate the final image. And the advantage of a cyclic acquisition in this form is that it can make optimal use of the brightest channels in a fluorescence microscope. There are other methods using mass spectrometry that I won't discuss today. And you see over on the right then sort of the result of something like this at intermediate resolution. And this shows you just a couple of channels. And the sort of cyclic whole slide data acquisition can go up to 120 channels. There's a certain amount of sort of bragging rights on these things. But in practice, we find that between 20 and 40 channels is often the most informative for the types of tissue specimens we're going to look at today. And I show below the fact that these types of instruments, or this type of method, can actually be implemented on an incredibly wide range of instruments. And we're going to draw on that capability as we tease out our initial 6 lessons. So I'm going to refer many times to that data from the Cell paper, in which we have whole slide imaging. And what we've done here is taken a cohort. It's now up to 75 patients who have stage 2-3 colorectal cancer that resulted in resection. This here is showing you a cyclic immunofluorescence image at about 25 plex. A single image, remember, is going to be around 10 to the 6 cells. This whole data set is around 10 to the 8 single cells. And these are pretty big images. So that represents part of the challenge here. And you see the 3 ways we sort of represent the data, and that will come up again. This classic H&E morphology, which is absolutely critical because it links into 125 years of experience with histopathology and diagnosis. The multiplex tissue image, and again, you see only a couple of channels here. You'll see those bright white spots, and you'll think, what are those? Those, for example, are where B cells are highly concentrated. We'll talk about that more in a moment, so-called tertiary lymphoid structures. And then you'll see the single cell representation at the right here, shown as a t-SNE plot, where each of the different colors here is indexed below and represents a different cell type that we can discriminate and then map back onto the sample. One of the things you'll notice in a tissue image, and is both, I think, quite exciting, but also quite challenging, is the extraordinary range of spatial scales. So one thing I'll tell you about today, and here we're looking at a picture of a normal human colon at very low resolution on the top. So notice that in that case, you're looking at several square centimeters of tissue. At intermediate resolution down below, and you'll see those little kiwi-like structures. Those are the cross sections of the villi in the normal colon, the normal colonic mucosa. And then on the right, you'll see a very high resolution reconstruction we've done of a single macrophage. And tissue imaging, it's turned out, even in archival specimens, we can see things right down to the level of the diffraction limit of the microscope. You can count chromosomes, you can look at the intermediary filaments. So all those things we could do in tissue culture, we can also now do in tissue itself. And that can even be archival tissue. And as I mentioned before, that's done by getting many images, either over multiple channels, but critically to get the pictures of this size, which are up to 100,000 pixels square, you're going to need to have many images stitched together. So here's just a quick representation you're going to see on the left of a movie. This is this 3D reconstruction that we did. We did the 3D reconstruction both to understand something about colorectal cancer, but I'm going to tap into it today from the purpose of thinking about the spatial scale of individual elements. The colors here just represent some of the classic elements. Bright pink are the epithelia, normal on the right, tumor on the left. And then we also do, typically in combination with this, micro-regional transcriptomics, so a sort of form of spatial transcriptional profiling in order to get the gene expression profiles. Now, one of the things we learned in this, and I'll just give you a quick representation, and this represents lesson 1, is that many of these structures are actually much, much larger than we ever imagined they were before we did 3D reconstruction. So on the left, you're going to see a classic view of one of these B cell, immune cell rich structures, the tertiary lymphoid structures, those bright white dots in the middle picture. You'll see it on the left on H&E, and then down below, you see it in an immunofluorescence image. You can see the resolution here. We're looking at a couple of thousands of cells. And they tend to be thought of as though they were little isolated pools, and that's what you're seeing on the very right hand. Just one tissue section, a couple of square centimeters. We're plotting out with a computer the locations of all of these tertiary lymphoid structures. And we were able to enumerate in this set of specimens, 3D specimens, we have enumerated several hundred of these, actually I think almost 1,000. However, when we actually stacked all of this up and did a 3D reconstruction, which you're seeing now on the left in rotation and on the right in a single view, is that in fact these things that look to be small isolated structures are in fact large interconnected elements. And that has gone on to be true of almost everything we've looked at in these tissues. Their interface with the colonic mucosa, the organization of the immune system, the way in which the invasive boundary pushes into normal tissue. So things that you might have thought were on the scale of hundreds of microns in these kind of three-dimensional views are really on the scale of millimeters and centimeters. And we're going to see that is incredibly critical to the way we think about sampling this type of data. So the most common thing you'll find in the literature for trying to get access to the features, particularly of tumors, that might be relevant in thinking about, let's say, a diagnostic setting or in some sort of spatial biology research setting, is going to be use a tissue microarray. And a tissue microarray, you see one here that derives from many different tumor types. More classically, you'd use many different patients from a single tumor type, but it has the great merit that you can put many different patients on a single block. And here you can take your 10 to the 6 cells and actually get a whole bunch of different examples of that. And so this has been, as I say, the most common way. And there are a whole series of recent manuscripts that suggest that you can get diagnostic insight from this, where each core contains around 3,000 cells. So 3,000 cells seems like a pretty decent number of cells. If you were doing single cell sequencing, you might think you were sort of on the verge of statistical significance. However, let's now go back to the whole tissue image here. And what we're going to do is imagine that we sampled this tissue image with a piece that was the size of really the largest commercial tissue microarray you might ever get. And that's what you're seeing in yellow. Alternatively, you could actually look in a lot of the popular mass spectrometry methods. That would be what you see here in green as a square. So just to show you what we're doing, we're imagining, and I just put a couple of colors up here representing 2 different cell types. We're actually going to slide a little tissue microarray around, and we're going to ask what happens if we sample at that spatial scale. One of the first things we realize is that things are actually, as I mentioned, pretty big. So a typical tissue microarray core radius, which I've shown here as on the length scale, is right around the relevant size of most features that we see in these images, here represented in the most primitive way, just by a single marker. And we look at a bunch of cores that we could pull out. And as I said, we could do that both virtually, but actually the pathologist in this case kindly went and actually extracted a bunch of real ones for ourselves. So we have both. And then we're just going to take those cores and we're going to go through and we're going to count something simple, like the number of keratin positive cells or FOXO-P3 cells. And you're going to see that on the right. And what you'll see is those TMAs, here called the virtual TMA or VTMA, have everything from 0 cells in them to nearly 90% abundance, whereas the true number, if we had sampled the entirety of that specimen, is going to be around 45%. And so this turns out to be a concept that was known all the way back to Lyapunov and Laplace, and it's called sampling under correlation. And the concept is if something is pretty big, you need to take a picture of it that's several-fold bigger than that item in order to be able to get an effective determination. So lesson 2 here, and I realize I've gone fairly quickly and you can see a more complete exposition in our manuscript, is that if we have large things and many things in tumors and tissues or centimeter scale, we can't figure out what we're looking at by looking at pieces that are a fraction of a millimeter in radius. And there's simply no statistical power in that. And this is an important lesson because it tells us now that we're going to have to lean into this concept of whole slide imaging. And whole slide imaging comes with a cost, and that cost is namely bigger file size and greater complexity. And let me give you one other easy to understand example of this, is we tend to think of tumors as, particularly from spatial transcriptional profiling, as being positive and negative and critical elements. And here I'm just showing you 2 sort of semi-randomly chosen colorectal cancers from our cohort. On the left, we're looking at, in a yellow-blue color gradient, we're looking at the concentration of the epidermal growth factor, which is a critical oncogene in this tissue and actually a therapeutic target. In the right, we're looking at a histone modification that's involved in epigenetic regulation. And you'll see quickly that these are actually graded phenotypes. And in fact, and if I just show you a couple of cores here, you can see that each core can actually sample really without any intention. And these were chosen actually by real pathologists to be representative of the tissue. It can be a 10 to 100-fold difference in level across any such tissue. And these microarray cores recapitulate that, but unknowingly. And so I think this really all relates to this critical feature of intratumoral heterogeneity. All of this is sort of remarkable from a biological perspective, but my point here today is its critical impact on the way we think about actually doing imaging experiments. Now, it turns out that spatial analysis in a diagnostic setting already requires whole slide imaging. So we sort of knew this along the way. The FDA and I believe the EMA are going to make that a regulatory necessity. And the fact is, no pathologist is allowed actually in a cancer staging setting to look at a TMA to make a diagnosis. Another thing that's really interesting is the value of very high resolution imaging. And I alluded to that before. And here you just see a couple of pictures, and I'll show you one more depiction in a second, of a relatively high resolution imaging we've done from an archival specimen that had been sitting around in the pathology department for a few years of the invasive boundary of a melanoma. And as you might imagine, one of the things we're most interested in that setting is the battle between the immune system and this emerging tumor that's trying to invade. This is an early stage melanoma. And here you can see, not only can we look at individual cell types, here's a T regulatory cell, and we know it's a Treg based on the spectrum of markers, some of which are shown and some not. We know here's a tumor cell, each of these is labeled, and then the CTL is a cytotoxic T lymphocyte that's actually attempting to kill this tumor cell. And we know that that's true because we can look directly at the formation of a T cell receptor mediated juxtacrine junction with a tumor cell. So high resolution information of this type allows us to access features of protein distributions which are representative of function. And that strikes us as very critical because we only get a single fixed time point. We can't in a human setting go in and do genetic experiments, of course, but if we can enumerate at this resolution, we can actually find things that are representative of the interactions that we know to be critical. And here's just one other example. Here we've actually rendered this in software. This is a high resolution 3D example. And what we have here, interestingly enough, is a T cell here marked with T that is PD-1 positive. So that means it's an activated T cell, and it's activated but being suppressed here by this macrophage that's sitting off to the right. There's an M, and it's reaching an arm around and doing a suppression event. And most of you will recognize that this type of PD-1, PD-L1 interaction is the primary contemporary target for cancer immunotherapy. So being able to enumerate these interactions and show where they lie in a cell is actually highly informative with respect to therapeutic mechanism. The other thing then that derives from that is this critical role of resolution. And here are just some pictures that I've drawn from the literature from a couple of different spatial profiling platforms. I'm not going to identify them for you, but you can find them out there. And then on the right, one that we've collected on a really high resolution microscope. And you can see these are all at the same resolution. So you can get sort of a sense, once again, of what you can capture at resolution. And I'll just pop up a view here. This is from a Zeiss LSM 980 microscope. And you can see all these sort of really interesting features, which we would like to now be able to enumerate and characterize. LAG3, a critical checkpoint protein here, is polarized. We can see the way CD8 is on the membrane. We can directly quantify nuclear pleomorphism. So resolution then is one of the critical things for us to think about as we go forward in spatial profiling, not just most genes you can get onto a membrane, but instead the resolution with which you can do it. And by way of information, the lateral resolution in this image you're seeing here is about 140 nanometers. Now, one of the greatest challenges, and I'm almost getting to the end of these lessons, is really image processing and analysis. That actually in our lab represents one of the hardest single tasks You see a bunch of the papers that we've published here as part of H10. These are all open source, and they're all going to be represented onto the Cancer Research Data Commons with general use. But despite this, and despite our having been happy to get these things published in good journals, it's worth noting that the way we do single cell analysis in contemporary spatial profiling is as though we were using a microscope like a really low quality flow cytometer. And that is to say, we integrate the intensity. We find cell nuclei in this representation in the left of a typical picture. We find cell nuclei. We integrate the total intensity of all the marker space. We plot that on some sort of a gate, again, as though we were flow cytometrists. And then we use a tool, panels 3 and 4 here from Cell by Gene from CZI Science. We use some tool to then represent this in high dimensional space. But I think as I've shown you today, that is definitely not cells. They're not little spheres. They don't live in there. And typically for a microscope, the information lies in the morphology, not in the intensity. So I just want to give you that last caution. And there are now a series of new methods coming along. And I'm just giving you one example where the machines learn directly. Here, this is a form of machine learning known as a variational autoencoder. You can see on the left what we see as humans in this picture, and on the right what the machine has learned. And so this representation will, or in this case, the machine doesn't go in and try to quantify a B cell based on having, say it's a flow cytometer, so much of one signal, but instead entirely on the morphology. And just to give you a hint, this will come out, I think, over the next couple of years. We'll get these into routine use. Here's just an example, slightly silly, but of putting a t-SNE plot type representation where what we're looking at is not the intensity of the signals, but in each case, the morphology of the cells and its immediate neighbors. So just to give you that sort of future look at where we are in having for the software. And then the last thing to remember is a lot of what holds tissues together is not, in fact, cells. They're held together by all sorts of acellular structures. And here's a picture of, again, in early stage melanoma in the skin. And you're going to be struck not by the cells, which are those little speckles at the top, but by these bright fibrous structures in yellow. And that's actually the collagen. And we can image that directly in this case using a special form of imaging called second harmonic imaging. But this is going to come up again as we look to multimodal imaging, is don't be misled, particularly if you're doing primarily transcript profiling and thinking you're seeing the majority even of the mass of a cell or of a tissue by looking at the nuclei. And these are the structures that get disrupted in disease and are critical aspects of our overall analysis. So what matters then? What can we come away from saying from this set of studies? What matters in spatial-based profiling? So much of the field is focused on plex, not unreasonable, because with more plex, we get more discrimination of cell types. I've shown you the value of 2D versus 3D, made the point that microscopes are not flow cytometers. So there's an entire aspect of sort of the intersection between resolution and depth of field that we're only starting to appear. Statistical power says you can do antibody validation on tissue microarrays, but you can't do statistical profiling or spatial profiling. And then finally, I think most people would agree there's an additional advantage to bringing in other modalities. And we do that both with H&E, as you'll see, and also with spatial transcriptomics. So there are going to be trade-offs in this space, and no single platform is going to be able to do all of these things optimally. But I think it's important to be aware of where you live in this sort of set of somewhat overlapping, but also somewhat orthogonal technical requirements. And so that in mind, I want to introduce a new approach that we've developed and that is in that bioarchive manuscript. And it attempts to find a particular solution to that optimization that optimization problem. And this has been commercialized by Raresight as an Orion workflow and came out of that National Cancer Institute SBIR grant. And the idea was here to get away from a cyclic acquisition process, to make it simpler, less expensive, more automated, and honestly also to make the data analysis somewhat easier. This is going to be immunofluorescence imaging. We're then going to try to separate all of the one-shot, what we call one-shot immunostaining here, into separate channels. And this is an idea that's been around for a long time, but has really changed in the last 2 years, 3 years with the introduction of much more sophisticated solid state lasers and optical elements. And then you're going to get a multiplex fluorescence image of the type we've seen here. We're trying to go for the best possible resolution consistent with good scanning speed. And then we're going to do a same slide H&E stain. And as we'll see, along the way, that seemed like a good idea, but that has emerged as one of the most interesting features of this type of multimodal imaging. So imagine in step 4 here, we're seeing sort of molecular precision, which I think Laura referred to in the beginning. In step 6, we're seeing 150 years of histopathology. And of note, in a research setting at least, or in an academic setting where you're doing this on your own, you can go ahead and cycle this and get higher plex if you desire, obviously at the cost of slightly higher complexity. So here's sort of an example of a multimodal image. And at the end, I'm going to show you a little animation that we'll soon make, we will release with the manuscript itself, that you can play around with. And on the left is 5 channels from the fluorescence image here. You can begin to get a sense of the resolution we have. Pretty good. We can see cell morphology. Can't see those super fine features that we could see in a laser confocal microscope, but pretty optimized resolution. In the middle, we can see the H&E of exactly the same cells, not the adjacent cells. Adjacent section has been true until now, but actually the same. And then you get this remarkable capacity to overlay them. And when you look at the two, you realize, my goodness, the pathologist says, here are cells I would never have appreciated were there from the molecular image. But as I'm going to show you in a moment, there's a lot in the H&E image that we can't see in the fluorescence image. And that was sort of unexpected. The other thing that you can do with a kind of multispectral microscope is you can unlock all of these forms of endogenous fluorescence. And there's a whole literature of this dating back to the 70s and 80s when microscopes were a little less sophisticated than today. And what you're seeing on the left at relatively low resolution, and then really labeled the exact same resolution in the same section, labeled on the right, is the cross-section of a blood vessel. And you can see all the blood cells, the red blood cells inside, actually based on a fluorescence signal. And critically, you can see this elastic lamina, the thing that actually holds the blood vessel together. And that shows up as bright blue. And what we're learning now as we go through these tissues is all of these kinds of structures that we can see in endogenous fluorescence. Classically, the problem was if you didn't have molecular markers, you didn't quite know what you're looking at. But the combination here of molecular markers, and you're seeing 2 CD31 for endothelial cells and smooth muscle actin here for the muscle, you can very quickly appreciate features of the morphology, no antibody needed. So a sort of a free channel, if you want to think about it that way. One of the things we noticed when we had the dual images is that there were a substantial fraction of cells, and you can see the little plot here in the upper left-hand panel, it averaged around 20% of cells, for which there was a clear nucleus, but we had no assignment in our high-plex image. And that was true even when we actually took these images through many more cycles of imaging. And we wondered what those were. And in this case, we were able to pop over to the H&E channel. And then we used a machine learning algorithm that we got from directly actually off of previous publications. And we asked that machine learning algorithm to identify the cell types in H&E that were invisible to us in this more expensive immunofluorescence world. And you can see the initial assignment on the right, confirmed in many cases, but not definitively by pathology reinspection. A lot of them were stromal cells. That was most abundant. And in fact, we have no definitive stromal cell marker in here. It's hard to get one for each tumor type. There's also smooth muscle, and adipose are very abundant. Smooth muscle, you have many nuclei per cell, and they're very, very elongated, and they're essentially missed by all segmentation algorithms. And then we have a couple of other categories, including debris. But what we learned from that was that actually the combination of H&E and IF imaging took us from about 80% of cells identified to greater than 95 to 98% of cells. And that actually is a huge strength when we begin to move forward to actually understanding determinants of disease. So we thought we would actually test this method in a diagnostic setting by comparing it to some really revolutionary work done by Jerome Gallon and his colleagues in developing a test known as Immunoscore. And critically, in Immunoscore, as part of a huge validation project, what they wanted to do was quantify the extent to which the invasion of a tumor by lymphocytes would be prognostic of disease outcome. And that would be used in several settings, but initially colorectal cancer was the setting of choice in order to determine whether patients did or did not need adjuvant chemotherapy. And those patients who don't actually could be spared a highly cytotoxic treatment and nonetheless have good prognosis. And the way this was done was they took tumors and they divided them into the center of the tumor, CT, the invasive margin, IM, and then they counted the number of T cells in there. And you see CD3, CD8 doubly positive cells are a particular subclass of T cell. And that was done with classic immunohistochemistry, although it was digitized. And you can see that's what we call the brown stain here. And then an immunoscore could be calculated based on the relative abundance of imaging in each of those compartments. And so now with having a digital method like as the one that I've shown you, now we've got a 20-channel image. We've got that on a censored set of 73 colorectal cancer patients, all at the time of initial diagnosis. It's really easy in a multi-channel image to actually find all of those features that were identified for immunoscore, and we can find the invasive margin computationally. You just see that laid on for one tumor here. And then we can calculate the abundance of all of those cells and really recapitulate that sort of revolutionary work that was done before, and we actually achieved exactly the same hazard ratio. So not remarkable, but just showing that it is in fact possible to fully automate in a single image all of what previously took multiple sections. And there's actually a whole sort of emerging branch of our research that actually finds those applications in contemporary pathological practice. But of course, oh, and I should say, actually, for those of you who are not so familiar with this, what we're looking at on the right actually is a Kaplan-Meier survival curve. So survival probability in this case is progression-free survival. So whether a patient after initial diagnosis and surgery represented with disease. And the discrimination we're looking for in this biomarker is between a high score, in which case you would end up with slowly progressing disease, so good survival, and more rapidly progressing disease. And the hazard ratio, in the simplest way of thinking about it, is really the difference between those. And it's about a 5-to-1 difference, which would make for a really high quality biomarker. And indeed, that's why immunoscore is important, particularly in Europe. Now we can improve on that because we have a lot more features in here. So we've actually measured 18 different features, of which we have 14 immune, and then there are a bunch of tissue features. And we can actually go through and do a series of ways to actually find different combinations of these that would be more or less effective. And we do that in a kind of penalized way because, of course, there's a kind of multi-hypothesis test that's being done here. And this just shows you another example of an additional molecular feature that we found. And we can find hundreds of ones that perform better than immunoscore. Here's just showing you one example of that. You can do a cross-validation shown on the right. I won't go into the details of the statistics, but you can see we have substantially better performance in this case. And that's not surprising because we're measuring additional features of those tumors. And as I wrap up in a few minutes, we'll look a little bit into what some of those features might be. But the other thing you can do now when you have enough features like this is you can directly apply a machine learning approach to simply find predictive elements in the image or in the features of the image directly that relate to outcome. And here we've done that using a method called latent Dirichlet allocation, or spatial LDA, and you'll find references to that in our paper. And the idea is to find features, combinations, and let's look at the bottom one down below in red here. It's a whole bunch of CD20 cells with spatially proximate CD3 and CD45. And if you look this up, you'll see that actually is a tertiary lymphoid structure. So that is exactly the same structure we were looking at before, and it's long been known that having more of those is associated with better prognosis, and that's in fact what the machine found here. But what I want to draw your attention to are things that are not immune related, because we've just been looking at the immune things, working from Galland's revolutionary work on this, and now we're going to look at cadherin versus pan-cytokeratin. And the interesting thing is you see that the ratio of these two measurements here seems to be prognostically significant. And yet it is typical of machine learning models of this type that you don't get deep insight into what you've learned. I couldn't tell you what these were. And the neat thing about having same side H&E images is you can unpack this again. So we can take this back to our pathology team, seen here imagining, this is actually at Johns Hopkins, imagining they're actually looking at this directly and look at the interpretability. And that unknown machine learning model in image space looks like what I showed you at the top. And this is an incredibly invasive form, locally invasive form of cancer associated with discohesive growth, as it's known. We think this is an EMT-like transition, and we'll learn more about the molecular features in the future. But the neat thing is having this kind of multimodal image allows us to learn on features the way modern machine learning methods do, and then pop that back into a kind of biological space in order to interpret that. And in general, we think in these diagnostic settings, but clinically for research too, that it's this interpretability of these models that's going to drive progress. So if we combine those two, the machine learned feature that came from tumors, all the stuff that we can learn about the immune system, you can see we can get about a 20-to-1 discrimination and progression for this patient population. And so that's actually a sort of, I think, a pretty interesting proof of principle, and allows us to now begin to think about advancing this type of thing into a care setting. But let me end here with a very important caveat, which is what I've shown you today is a single center retrospective cohort study. So it says CRC is here. That's the study I'm showing you today. The minimum for this to be considered even for a testing setting in patients is going to be a multi-center retrospective cohort, at least 2 to 3 times more patients. And then eventually, if you wanted to go and sell this and somebody wanted to pick it up, you'd have to take this all the way into regulatory validation. And that's a substantial downstream development. But from our perspective as academicians, what we're trying to do is get this into a hospital setting as a CLIA-compliant facility, can actually do this without FDA approval. And we think that's about 1.5 years away if these results hold up. And the last little bit that we're doing is a lot of integrated analysis with genomics. So it's sort of interesting to think, well, there are genomic features in these tumors, and I've shown you image-based features. Show me a way of comparing them. And so what I hope we'll be releasing a little later this year is the possibility of looking at everything I've shown you today inside of cBioportal, which is the dominant platform for doing cancer genomics, both in a research setting and also in a translational setting. So, so hopefully that'll be in there, and then you can compare them. And I'll just give you one little tidbit of this, is that in general, we find that the things that are image-based are actually substantially more powerful than any single feature that we pull out of a genomic analysis of that type. So my final slide here is just sort of a little animation from something we'll be releasing fairly soon. This is an in-browser look at multimodal images, and you'll be able to pop this up. I intentionally just took a screenshot of my own browser, and what you can see is here we are sort of scanning between an H&E image and a multimodal image, and we can then move this around. There's probably some bandwidth issues going on here, so for those of you who can't see it, we'll pop that up and put it on the website for as well. Yeah, sorry, there's an imaging problem here, but this is sort of an image trying to show you what you could actually get from multimodal image, and then pull that forward in the future into these kind of research settings. So I want to thank you all for your attention today. Thank the key pathology collaborators who made this possible, particularly Sandro Santagata. Members of the lab I've tried to recognize along the way, and we've been critically supported by a much larger group of collaborators here at HMS in Europe and across the United States. So thank you very much for your time.
Thank you very much, Peter, for that presentation. It is now time for the question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. Okay, so we will come back into the studio. Thank you once again, Peter. Can I just ask you to adjust your webcam slightly? Thank you. Just to get us started, I would like to ask you, How long did it take you to scan the samples that you used in your study?
Sorry, just getting myself adjusted here. Yeah, it's a great question. It's not it's so remember, this is the first use of this technology in the wild, and so as a consequence, this kind of thing played out over a couple of months. But as a practical matter, we've been able to do a cohort of the same size in just over a week. So scanning these each slide takes a couple of hours. And as long as you have some enthusiasm for staying there with a machine, this will be automated in the relatively near future, I think. As long as you do that, you can actually build out a cohort of 40 to 70 patients, I think, pretty realistically in under a week. The harder part, honestly, is the data analysis and scientific interpretation. So that actually will be, the next challenge.
Right. And so actually the next question asks how long it took you to validate the panel before.
Yeah, so panel validation is obviously a pretty critical task. We did this both in-house with our pathology colleagues, and we did it in collaboration with Raresight. The typical way this is done, actually, so it'll be interesting to see how is this done in a CLIA setting. These are almost all commercial antibodies from other vendors that have been widely used. And the typical standard is a human standard, where you take an antibody and you compare it between something that's been in historical use and something that we're now going to do. And so an antibody panel of this type, validated to sort of kind of pseudo CLIA standard, CLIA standard is the clinical translational standard, takes about 3 months of effort. So it takes a while to build these panels up. As you build up the antibody panels, of course, you have them. One of the surprising things that has emerged from that, however, is modern antibodies from modern vendors are often monoclonal rabbit antibodies. And they're much more effective than the historical monoclonal mouse antibodies. And yet the clinical standard we're comparing to are often polyclonal antibodies that were put together 20 years ago. So actually, it's not obvious to me that the research standard is lower currently than the clinical standard. So you'll see a lot of movement in that, and the antibody vendors are very, very tuned into this. But I think you should expect that an antibody panel takes a while to correctly validate. But once there, you can use it fairly routinely across multiple tissues.
Okay, thank you.
A longer answer than you were looking for.
It was definitely longer than I thought it was going to be.
It's a big concern in this space.
Our next question asks, what are the features of precision microscopy platforms that are necessary for your work? So what are you looking for from this type of platform?
Yeah, so I mentioned that was sort of meant to be the essence of 6 lessons, right? So it's sort of, spatial scale. You've got to probably be whole slide. Remarkable when you visit your friendly pathologist, how often a human tumor takes many tissue blocks. So even if you're looking at a whole slide, you're still subsampling. Resolution is pretty critical, I would argue, because that's how you see cell biology in a tissue, and that's how you think about mechanism. And then I think I mentioned already, the value of plex, et cetera. So it's those 6 features that I mentioned that we see ourselves trading off, and different approaches could trade them off differently. And in a setting that we were pushing, which is going to be relatively high volume, because you want to get your first question, many specimens through, you're going to look for whole slide imaging that can be done efficiently.
Mm-hmm. Okay, thank you very much. How do you manage inherent autofluorescence?
Yeah, so there are 2 answers to that. One of them is, so all tissues have autofluorescence, and it has been the bane, actually, of immunofluorescence application in tissue. In the last few years, what we learned was actually you can knock that down by at least 100-fold by tissue bleaching processes. So essentially acid or base catalyzed acid or base catalyzed oxidation. And so that actually already makes things much, much better. In the instruments that I showed you today, we actually use it as a feature. So autofluorescence in many cases is a very valuable feature. It can tell you where are the melanocytes and melanoma. It can tell you the elastic lamina. I showed you a picture around blood vessels. So we extract it as a feature, and then we can also then subtract or account for it in all other images.
That's interesting. Yeah, to use it as a feature. It's really interesting. Our next question is about panels again. What do you think is the minimum and optimum number of markers in a given panel?
Yeah, so the answer is that there's an incredibly effective set of methods that actually are in use in clinical settings that use 6 to 8 channels. And so I think a lot can be done with lower channel number than people imagine. However, we judged in this case that a panel around 20 was what we were optimizing for. And if you want to balance that out, a panel of around 20 is about 12, 13 markers to get the major immune system. And then you have to get the tissue architecture. And then let's say you were looking in a therapeutic application, you'd want the drug target. So that was sort of what we were aiming for in this study. You can then go on, always at the cost of greater complexity and greater uncertainty about antibody panels, to get many, many more. So the sweet spot for sort of high-plex imaging, if I had to be I'm pulling this really just out of our own experience, is around 60-plex. The sort of sweet spot for really efficient imaging across many specimens we think is between 18 and 20-plex.
Okay, thank you.
And actually, I'd just say, Sarah, what's interesting is that is surprisingly close to experience with high-plex flow cytometry used for similar types of specimens. It really really key places around 20 to 26 markers. And here we have H&E, which we're sort of counting as being the equivalent of 6 to 8 fluorescence markers once we get the machine learning algorithms fully working.
Great. Yeah. I want to move on then to a question which asks whether you can do the same section H&E with SciSif and how the image quality compares between the Orion method and the SciSif method.
Yeah, so what I would say broadly, and the answer is yes, you can with SciSif or a cyclic method. But what we observe is that the quality of the H&E image, and implicit in your question is recognition of the fact that H&E imaging has to be done after all fluorescence imaging is finished. And the reason for that is that eosin is so fluorescent, it just blows out all other possibilities. So therefore, it has to be done at the end. And every cycle damages the tissue more with respect to H&E. So the trick, in a sense, with Orion is a protocol that's minimally damaging for subsequent H&E. If you did one round of SciSif, you'd be relatively happy with the H&E. But as you go to deeper rounds, and we find that's true with cyclic methods in general, as you go to more and more rounds and you collect that high-plex data, you get worse and worse H&E data. And so here we were aiming for something that a pathologist would call diagnostic grade. And that's really the advantage of a one-shot approach.
And that kind of actually answers another question that's come in, which is about how easy it is to apply the cyclic procedure repeatedly, which is you can.
Yeah, so two answers to that. One is there are many vendors out there actually use cyclic data acquisition routinely, so that's been commercialized. In our implementation of SciSif, we don't find it particularly difficult at all. You just have to know some basic microscopy, but it is relatively slow and it requires a fairly high level of skill. On the other hand, kind of one-shot imaging is something you could imagine putting into a core facility, for example, or in the application I discussed today, into a diagnostic setting, and is therefore easy. We did show in our manuscript that if you got your 20-plex image and you wanted some more, you can go ahead and cycle with Orion. You can actually do another channel, another round of potentially the same scale, and you can also exit from it into other cyclic methods. So you haven't reached the end of the road once you've done your first imaging cycle. But as I mentioned before, the more sort of manipulation you do of the specimen, the more cycles, the less the H&E component is going to be, sort of acceptable in this kind of diagnostic grade.
Thank you for that. Our next question asks about the spectral extraction limit in markers, and it asks whether the spectral extraction limits the dynamic range of immunofluorescence imaging per marker. if there is a comparison between Orion and SciSif in terms of spectral extraction limitations, and what that impact would be on downstream analysis. And the example they've given here is GMM-based cell phenotyping.
Yeah, it's a great question, and the answer is, we're going to have, actually, for almost all of these tissue imaging platforms, people who are asking these questions will note there is no published rigorous comparison the way we had for, say, single cell sequencing over the last few years. So I think getting to that is going to be really helpful to see these platforms working together on a single set of tissues. We were funded by the National Cancer Institute, in fact, to do that study as part of HTAN. But for the most part, we never managed to get specimens from enough different methods and vendors to actually do that. I anticipate someone will in the near future. Our own experience, we directly compared Orion to SciSif, and the data will be available in that paper, freely available through the HTAN website for reanalysis. We did not notice any reduction in dynamic range as a consequence of spectral extraction. But there are many features of these microscopes that play into this, and the trick for, and here you'd really have to ask the experts at Raresight, the optical engineers, but the trick here is really taking this concept of extraction and implementing it on a modern set of hardware in a way that really wouldn't have been feasible just a couple of years ago with all this new digital light processing type elements. So from our initial perspective, there wasn't a compromise.
Okay, great to know. Thank you very much. Our next question is about single slide images coming from, which are representative of just a part of an entire tumor. And the question asks how you can be sure that the single cell section image reflects the tumor. And you did address this a little bit, but perhaps you can expand and clarify.
Yes, no, it's a great question. So, I sort of spent some time and put up a little equation there to sort of go after the concept of doing spatial profiling in tissue microarrays. But as you're correctly pointing out, a large tumor resection could actually span many tissue blocks. And so it's not uncommon for a pathologist to scan those blocks fairly quickly, and then look at a subset of them in greater detail. So we're not able to overcome that other than to look at many blocks. What we think in the long term, and this is sort of a general commentary on the whole challenge of doing sort of proteomic or high-plex type analysis on any clinical specimen, whether this is single cell sequencing or not, is the number of specimens, essentially the number of patients that would be used in a simple study that was H&E based would today to be published would be 500 to 1,000 specimens. That exceeds by an order of magnitude anything that anyone has ever attempted with high-plex methods. And single cell sequencing would have the same challenge. So our current understanding is the way we're going to overcome this is by integrating at a machine learning level analysis of large cohorts of H&E specimens, and then very carefully curated cohorts of specimens that undergo Orion, for example, or other high-plex imaging. And so on one hand, we'll leverage the molecular depth. I think you referred to this in your introductory comments that you get out of molecular profiling with a minority of the samples, and we'll get at sort of statistical significance with respect to cohorts by doing relatively simpler methods. And in that regard, you could imagine taking something like Orion and just reducing even further the number of antibodies and making it ever more efficient and ever less expensive. And so playing that off in terms of both statistical power, which I discussed, and more conventional sort of power in predicting, say, an outcome or a feature is going to be a critical aspect of this research over the next few years.
Thank you very much. And I think probably our last question for today, it asks how far these very large structures affect disease outcome and require integration into the analytical workflow in your example.
Yeah, it's an extremely good question. And obviously, having now sort of discovered them, we now are going to have to work more on their significance. The one place I can point you to that is mentioned in the paper in our 3D reconstruction paper is not the TLS that I showed, the tertiary lymphoid structure today, but a phenomenon known as tumor budding. And so tumor budding is something you see at the margins of solid tumors, and it comprises little balls of cells, classically fewer than 4, that are seen to bud off. So imagine literally a little sort of vesicular structure that comes off the leading edge of the tumor. And you might imagine, and this is the theory, gives rise to metastatic invasion both locally and distantly. That is a prognostically featured with poor prognosis and is routinely scored. That was a structure that we discovered was not in fact little vesicles, but in fact is big fingers. So the leading edge of a tumor in the reconstructions we've done look like a series of fingers pointing out towards the margin. And what was called a tumor bud before was just a cross-section of the very end of these fibular structures. So that would be something I would point to as an example of something that's already known to be of significance from the perspective of disease. We just didn't know what it was. And now as we go in and do gene expression analysis, et cetera, we begin to appreciate that this is not at all similar to classic EMT that you see in a tissue culture dish. It's something very different. So I think in that way, and that's what we're most excited about, is finding those things already known to have significance from this 150 years of classical histology. And our job is now to give them the molecular description that will allow us both to understand what they are in scientific terms, but also to think about how they could be better attacked with modern therapy.
Well, thank you very much. That is where we are going to need to finish for today. We are aware to our audience that there are a number of questions that we didn't manage to get to, and we will get back to you offline after this broadcast finishes using the details that you've provided. So it remains for me to thank you very much, Dr. Peter Sorger, for being with us today and for answering all of our questions. Thank you.
Thank you very much. And if you go to bioRxiv, you'll see when the paper comes out for real. They track that very nicely.
We'll look forward to that. I would also like to thank our webcast sponsor, RareSight, and of course you in the audience for tuning into this webcast today. You will be redirected to a short survey. Please do take a moment to complete the feedback questions. And remember, you can watch this again at any time on demand at nature.com/webcasts. Thanks for watching, and I hope you'll join us again soon.